All right, hi everyone, welcome to our webinar today on propagating plants. Just a couple quick housekeeping things to go over first. We do have quite a few participants here today, still more trickling in. So we are going to save time at the end of the presentation to have questions. If you've been on Zoom before, you might know that there's a Q&A function just at the bottom of your screen there. If you do have any questions, if you could use that function, it'll sort of help us keep track of the questions we have coming in. I will be monitoring the chat as well, but Q&A does help us just keep track a little bit easier with this amount of participants. And if you aren't able to get your question answered today, you are absolutely welcome to send any additional questions to our email. It's at scemarketing@MacEwan.ca, and I will drop that in the chat shortly, and I'll also refresh all that at the end as well. So just a heads up, the webinar is being recorded. We'll be sending that out to anyone who's registered probably early next week. It's also going to be posted on YouTube in the coming weeks as well, so you can share it far and wide if you'd like. So we're going to start today with a land acknowledgement. Since this is an online webinar, we might not all be on Treaty 6 territory today, but Tan and I are both broadcasting from Treaty 6 territory, which is the traditional gathering place for many Indigenous people. We honor and respect the history, languages, ceremonies, and the culture of First Nations, Métis, and Inuit who call this territory home. The First Peoples' connection to the land teaches us about our inherent responsibility to protect and respect Mother Earth. With this acknowledgement, we honor the ancestors and children who have been buried here, missing and murdered Indigenous women and men, and the process of ongoing collective healing for all human beings. We are reminded that we are all treaty people and of the responsibility we have to one another. So with that, I will go ahead and pass the torch along to Tan.
Thank you, Lauren. Please speak up or in the Q&A, let me know if you're having any technical issues. Hopefully everything goes well. And thank you so much for deciding to spend a little time with us to talk about propagating plants at home. Feel free to use my email address if you have any questions after the webinar, any questions about plants, any questions about today's discussion. So what is propagation? Plant propagation is simply the process of increasing the number of plants. And we can see as a civilization many reasons we'd like to do this. So agriculture, of course, is the principal application of this idea. We can categorize plant propagation into two main categories: sexual and asexual propagation. So sexual propagation is what we know about the birds and the bees, where flowers are pollinated typically, and if successful, form seeds, and we can increase the number of plants we have by planting these seeds that germinate and form mature plants if successful. And this is one of my favorite topics. I think that the germination of seeds and the formation of adult plants is like many other things, but it's one of the miracles of life that I really enjoy thinking about. But what we'll focus on today is asexual propagation, which is the clonal propagation of plants, where the individuals, the plants that we produce from a parental plant is genetically identical to the plant we started with. And there are many reasons that we engage in this. One of the reasons, of course, is just to increase the number of plants of a particular kind, particularly something that may be rare or something that we have great favor towards. And this idea of asexual propagation is grounded in a property of plants that is also quite fantastic, which is that some of those cells of a plant are what we call totipotent, which means that they're undifferentiated cells, so individual cells of plants, that can become any other kind of cell. And the tissues in which these totipotent cells are found are called meristematic cells, or tissues, sorry. And this is sort of the basis by which we have the ability to asexually reproduce plants. And we'll talk about this more as we go along today. So what I'd like to leave you with today is sort of awareness of at least 3 basic propagation methods: dividing, cutting, and layering. And what I also want you to appreciate is that most plants can be propagated by different methods. Having said that, there's usually, as you get more experience looking at plants and their structure, there are usually methods that are more amenable for the particular kind of plant that you're interested in. And so this is something that you can experiment with and certainly look up if you are curious. But I'd like to introduce to you these 3 methods because they're the main backbones, I would say, of propagating plants. And I'll mostly focus on indoor houseplants just because we're entering that fall-winter season. And I was also thinking maybe if you start now propagating some plants, they might make nice little potted gifts for people in the holidays and so forth. So I'd also like to say, if you have questions at any point, Lauren's kindly keeping up with the Q&A. And so if something's unclear at any given point, please just type something up and I will try to get to it in real time. But we'll also have time at the end for questions proper. So what do you need to engage in plant propagation? So I think, like many hobbies, there are all sorts of fancy pieces of equipment and things that you could do to really enhance your enjoyment of the hobby, perhaps. But what I'd like to sort of start with is that plant propagation is really simple, and I think we could get most of the things that we need readily. So, for example, sort of the basis of this is having some sort of container or pot that's clean. And so you can purchase little green containers like this for about 50 cents, or I often use yogurt containers that I punch holes in with some sort of sharp implement. You need some sort of soil, some sort of medium. So this is, of course, assuming that we have a plant that we want to propagate. And then you need some sort of sharp cutting tool. And this does not have to be a fancy pruner or anything. You can just use often kitchen scissors, a sharp paring knife. You need something like clear plastic bags if you want to create a little dome, or you can buy little domes, and some miscellaneous equipment like a cutting board or pins. Now I'd like to step back for a second and emphasize a couple things that I would say are universal principles in successful propagation. One is that when we are propagating plants, a lot of times we'll be cutting them, or in essence, wounding them to some extent. And so having as sterile an environment as possible so that the plants don't get infected as they heal and as they regrow is pretty important. So if you have pots that you used before, you can just wash them out with soap water, maybe sterilize them with a little alcohol. Cutting tools, before you use your scissors or your paring knife, you can dip them in hot water, or also rub them down with alcohol. So this, what this does is just sort of enhances the probability that your plants will be able to grow back without getting infected in the process. Other things that I personally find helpful that may not be sort of as common, one are just sort of like large trays. So one of the things I find is that when I'm doing repotting and all sorts of things indoors in the winter and I can't get outside because it'd be too cold for the plants, I just end up making a mess in the kitchen and so forth. So having a little piece of tarp, newspaper, or just a fancy tray helps keep everything contained and sort of makes it, I guess, easier overall. A couple things that, if you're new to propagating, that you might find very useful are plant hormones. And so these aren't necessary to use, but when we do things like cuttings, you will find that having some sort of rooting powder enhances your success rate quite a bit. And the other thing, this is not particularly necessary in the propagation part, but lights, and in fact, like maybe detrimental, we'll get to that in a second. But once the plants are going, depending on the availability of light you have in your home, you can get really smaller artificial light systems that are quite reasonably priced and can be quite effective in helping your plants grow in our long, sort of dark winters. So when you're propagating a plant, as you're all familiar with sort of the plant's basic needs, one of the great sort of chemical processes on our planet is photosynthesis, where there is a conversion of energy from light into storage and chemical energy with the use of water, nutrients, and carbon dioxide. But what I want you to think about a little bit is when you are propagating a plant, in other words, let's say you're taking a cutting, you've changed something about that particular plant. And so, for example, if you take a stem cutting and you've lopped off any roots, then the plant's ability to take in water from the soil has been dramatically decreased because it no longer has roots. And as a consequence, the needs are the same, but the ratio of these needs changes because of the changed characteristic of the propagating plant. So for example, if you cut the roots, a lot of times what we want to do is keep the plant light, but not extremely bright environment, because when it's very bright, the plant loses moisture through its leaves, perhaps, or when it's very warm, and hence it goes into moisture stress because of the inability to draw up water into its structure. We can solve this in some ways by growing our propagated plants in water, but I just want you to sort of be aware that these things are such, and so temperature sort of has that same characteristic. Humidity can be quite important because, once again, let's say if you cut off the roots, the air humidity being extremely dry can desiccate a plant quite rapidly. But overall, I think when you make your change to the plant, it's quite obvious how you might want to compensate for the different needs. Okay, so what I'd like to start with is sort of just a review of the idea of transplanting plants, because it's sort of, and maybe just describe how I do it. I think there are many different ways to do it, but it is sort of a foundational sort of skill, very simple. Essentially, you're just like moving plants around or into another pot. But I just want to go through like why I might do this. So I have this plant called a Madagascar Jewel. This isn't a photo of my plant, but I have a plant like this. And what is sort of fascinating about this is it produces these little flowers that you can see on the top right-hand side there. And if you look at the bottom of a pot of the parent plant, as well as any pots that are nearby, you will see that there are all sorts of these baby Madagascar Jewel plants growing. And what's going on here is that the flowers, the plant can self-pollinate. So it doesn't need, I only have one plant, one adult plant in my home. And so it's not getting pollen from another plant. And so it's able to pollinate itself. It forms sort of these projectile seeds. By projectile, I mean they shoot out, I think at least half a meter. And why does the plant do this? Maybe just because any plant, baby plant, wants to get away from the parental plant, so it has its own space to grow. And anyways, I end up with lots of these little baby Madagascar jewels. So a very simple form of propagation, no thanks to me, is just I want to move the little baby plants to individual pots so they can grow up and have their own space to grow. And so what do I do? So the first thing I generally do when I'm transplanting a plant is sort of, if I'm not just doing it on the spur of the moment, and I'm actually planning this, I might water the plant the day before. And the reason I do this is when you disturb a plant, it might take the plant, and particularly if you're doing things like cuttings, you just want to keep the plant hydrated and sort of try to minimize any water stress by giving it water that you can store for the journey into a new pot. And then, as I mentioned earlier, I would have a few pots that are cleaned and prepared with some amount of soil. And so usually I like to fill the soil level, the bottom, maybe 2/3 of the pot, and then add more soil as I move the plant over. So the part that I think is sometimes the biggest leap of faith is removing the entire plant or set of plants from the old pot. And a lot of times what that means is like over a tray of some kind, literally just putting your fingers between the stems and tipping the plant over, sort of shaking the plant out. And I try to get the whole soil conglomerate into my tray and sort of like laid out so that I can see it from different sides and so forth. And it's kind of a leap of faith because the plant is now upside down and now out of its pot and on its side often at the end. But this gives us an opportunity to really examine what's going on inside the soil. And a lot of times it's pretty well organized. And so if you work while the Madagascar Jewel plants are very young, in other words, they're very small and the root system isn't that well developed, then it's very easy to separate the individual stems, and because the roots are not intertwined and sort of locked in. But if they are, you can sort of gently tease them apart, and certainly the less trauma you can cause the roots, the better, and the quicker they recover. But even if you cause quite a lot of damage, plants are pretty incredible in that they can recover. And then I would put each individual stem into a different pot, and suddenly you can see that you can have 20 Madagascar jewels very quickly. And so this is the idea of transplanting, essentially moving a plant from one pot to another. And we might do that in the process of just repotting. For example, why might we repot? We might repot because a plant is just getting too big for its pot. And here's the key: if you want the plant to be bigger, then you might want to put it in into a bigger pot. I think once you get into propagating plants, you'll find that one of the resources that you're quickly lacking is space. So the plants can get really big. In this case, these plants can get about 1.5 meters, 2 meters, if you give it the room to grow. And yeah, and you also might just repot because you want, let's say, a fancier new pot, or you might want to refresh the soil. Plants that have been in a particular soil for a very long time, there are different ways to amend the soil, but sometimes it's nice to just give it a new pot with new soil, where you, in the process of transplanting, you shake out as much of the old soil as you can, and you can use fresh soil as you put it into a new pot. So if we sort of extend this idea of transplanting, we get to our first sort of propagation technique, which is dividing. And so why might we divide? So dividing is a scenario where we can see, in this case with the snake plant on the right-hand side, on the upper right-hand side, that there is sort of a growth of rhizomes from rhizomes of the parental plant, I guess. And what you have to do is actually start breaking up the below ground parts of the plant in order to be able to move them. And in this case, what you're actually doing is you're really breaking up, but maintaining some root as well as some shoot, the green part, for each piece that you're breaking up. And so why would I have divided snake plant? So one of the reasons is what's incredible is that when you have a growing plant, there can be tremendous pressure on the inside of a pot. In fact, with one of the snake plants I had, it was just, it was able to crack the plastic pot. And so I had to change pots and divide it because the soil and water was just leaking out of the side of the pot because the plant, the force imparted by the plant growth literally broke the plastic container. So the process of dividing is very similar to transplanting in the sense that what we do is we take the plant, we, well, in my case, I would probably tip it on its side and sort of grab the base of the leaves and pull out the whole plant unit out of its pot, put it on my tray, and you'll see it'll be much more interlinked than this photo here, which is sort of mostly two-dimensional. And you sometimes have to get quite aggressive to be able to break up the plants. But the goal is sort of to imagine where the easiest pieces are to break off so that there's as little damage as possible to plants. But I have in the past where the root structure was so tightly bound, just taken a bread knife and cut right across the surface, and the plants that were made from that original plant were perfectly fine. So you can see what it looks like. I think it is easiest to use your hands to break up the plants and divide them, but certainly implements can be used. And if you're outdoors and you're doing this with perennials that are quite large, you might take two pitchforks and put them back to back and use that as leverage to split that root mass up. But in the end, it's pretty straightforward. You have a plant that's just smaller than the original, or you have plants that are smaller than the original plant, but they should all have roots and shoots. And so the process of propagating them is just very similar to the transplanting process that I described just a few seconds ago. So which kind of plants are readily divided? Some of the common plants that I've readily divided are the ZZ plant and Boston fern. But you can see sort of so the ZZ plant is the below ground structure is very similar to the snake plant, in that there's sort of these rhizomous structures that can be broken off typically. The Boston fern, you can see that the root structure is much finer, and in this case, a little saw was used to just cut through the soil, split it in half. Like I said, a butter knife, or sorry, not a butter knife, a bread knife works very well too for this purpose. And in each half, in this case with the Boston fern, can be put into a large pot, and you'll essentially have two Boston ferns at the end of this process. Another kind of very easy way to increase the number of plants is when you have offshoots or plantlets. And so what I'd like you to sort of appreciate here is that when you look at a spider plant, many people have, it's the plant is sort of speaking to you as to how it might be propagated. So just to be clear, the spider plant can also be unpotted and split up into large units. And that's great if you want two pretty large plants. But if you're just trying to propagate lots of spider plants, these little plantlets, so if you can see along the stem of the plant that you see little bundles of leaves, and these leaves can then be placed either in water, where you can see root formation take place, or they can be directly placed into clean pots with soil. And so we come to this point where I think people have different approaches and different preferences as to how they like to do this first stage of propagation. I often like to put them in water, not because it's better, but because I really just like watching the roots grow and I know that they're growing. But I know a lot of people who just like putting their plantlets, in this case, right into the soil because they don't want the extra step of transferring them from cup of water into the soil. Now, when you're working with water, and we'll talk about this with cuttings and so forth, one of the keys is to refresh the water with good regularity. I usually, when I'm doing something like this, leave the plants pretty close to my kitchen sink or something, and maybe every other day, maybe even every day, I'll just change the water because I'm paranoid that way. I think you could probably get away with every 4 or 5 days or so topping off because Edmonton air is quite dry. But the reason you want to change the water, just like when you're sprouting, if you're growing sprouts in your home, is just to keep the plant from getting infected by bacteria or fungi that may eventually float into your water and damage your plant. Soil is sort of magical that way, in that a lot of times soil is not inert, rather it's an ecosystem in itself. And for very complex reasons, a lot of the infections that would take place, let's say in water, don't necessarily take place in soil. And so that's definitely one advantage of choosing to use soil, other than beyond just the fact that it might be more efficient. So something like a spider plant is a great example where you have many options as far as how to propagate this plant. The plantlets are sort of the most obvious. The splitting is something we've talked about, and we'll also talk about it in the context of layering. I just want you to maybe become familiar with the idea that, so in one of the previous slides, I had shown you some plant hormones, and one of them is a cytokine plant hormone. And how you might use that is if you were interested in creating plantlets on orchid plants. So your typical home Phaneropsis orchid plants, when you have a flowering stem, you can sort of pull back some of the coverage on a bud. You apply this hormone, and what you find is that after around 4 to 6 weeks, you'll have little plantlets, or keiki as they're called with orchids, that form along that stem. And if you're interested in having more orchids, you can take those keiki off when they've grown enough, and they look very similar to these little spider plant babies. And so just to go back to why you might use plantlets versus splitting, one of the downsides of using these plantlets is that they're relatively small. Spider plants grow very quickly, so that's not really a big deal. But when you are working with something like orchids and you create a keiki or encourage the growth of a keiki, it will take you a while before you have a plant that's as large as the original parent plant. But it's a great sort of thing to be able to make more orchids. So now let's move into a different kind of propagation, which are the cuttings. And this really starts to, I think, illustrate the sort of the wonders of plant growth. Because what we're doing when we take, in this case, a stem and leaf cutting, what that means is we take literally from a parental plant, we just cut some part of the stem that has leaf on it, and stem and leaf. We often pull off some of the leaves at the bottom of the plant. We dip the plant, once again, optionally in rooting hormone to encourage growth. And you can either put it in directly into the soil or into water to let it form roots before you transplant it into soil. Now I want you just for a moment, I think this is just incredible. You have this plant, right? This cutting of a plant that has no root at all. There's just the shoot and with leaves and the stem, and somehow it starts growing roots and becomes a healthy, viable plant upon growing its roots. So what's going on? This is one of the, this is a intensive area of research. But what we can understand for now is that when you cut a plant, just like I think we associate perhaps maybe with animals more, being animals ourselves, but the plant is able to detect that wound. And so the wound is detected sort of with a cascade of hormones that are triggered by the actual process of damaging the plant tissue. And as a result, there's this signal that's created to the plant to sort of start healing that wound. And just as an aside, the reason that we want to emphasize using sharp tools is that when we cut a plant, whether it's with scissors or a knife, we don't want to mash that tissue up we're cutting, because then we're really damaging maybe too much of the plant, and it just makes it more likely that it gets infected. So a nice sharpened cut makes for, usually at maybe a 45-degree angle to increase the surface area. It is a great thing. And the other thing that's going on is that these hormones, so there's no central nervous system in a plant, but what it is able to detect because of a lack of hormones that sort of emanate from the root is that this plant no longer has any root. And hence the plant responds to this using some of its meristematic tissue enhanced by what's called a callus. So when the wound heals, so just like a callus we get from after we scrape ourselves, the plant forms a callus, and sort of wherever the meristematic tissue is near the callus, it starts to grow the roots and establish itself again, which is pretty incredible. So let me just talk about how I would employ this with hibiscus plant. So once again, in the case of a hibiscus plant, I would water it the day before. I would target a stem that was that grew relatively recently, ideally sort of within the last year or a few months of this process of cutting. And then you take your pruners and you cut at about a 45-degree angle. And the reason for that is that it's just like when we buy cut flowers from the grocery store and we put them in water, having a 45-degree angle increases the exposed surface area so that when we put it in water, it has the best ability to draw in water. We pull off the bottom leaves so that they don't sit in the water and rot. And of course, if you want to, you can dip the stem in rooting hormone. So let me just talk about that for a second. Rooting hormone comes in different forms. It comes in gel form, liquid form, powder form. Powder form is probably the most shelf stable. So I've used the same container for 10 years. And so I like the powder because I don't use it that often. But liquid form can be more active when it's fresh. And you essentially I always put a little bit of the powder into a separate little container or piece of paper because I don't want to contaminate the actual source of the hormone. And then I just literally dip the exposed area of the cutting into the powder and let it sit for a few minutes. And then put it in the water. And so in the case of the hibiscus, if you put it in water and once again refresh the water periodically, maybe every few days, you will get rooting. And in my experience, it does not take 2 or 3 months. It only takes a couple of weeks. You'll have a hibiscus plant that is rooting and that you can transplant in a pot in soil, and you'll have more than one hibiscus plant pretty quickly. And so let me stop for a second and think about why we use cuttings sometimes in the context of some of our food plants and maybe even sort of in the context of horticultural plants that we want to keep. So if you think about, I think one of the things that we've talked about in an earlier workshop is propagating something like an avocado plant from the seed that you find inside your avocado plant. And if you take care of it, so you might get it to germinate in a little cup of water, you plant it in soil, and if you're able to keep it going for a few years and you have good light conditions, then you will have a flowering avocado plant. But the problem is when we make plants from seed, that seed product is the offspring of a maternal and a paternal plant, right? And so the fruit that's produced from the seed that we buy from the grocery store, like an avocado seed, is going to produce a plant that is from the maternal plant that was part of the avocado crop, but pollinated by oftentimes like some other random plant. And that particular maternal plant happens to produce tasty, ripening avocados that we enjoy eating. But when you pollinate it with pollen from a plant that you have not selected, then you might get something that is almost inedible, never ripens, not necessarily rich and fleshy. And so why we take cuttings and why cuttings are made in a lot of apple orchards, I mean, universally in all apple orchards and pear orchards and grapevines and so forth, is you actually want to propagate an identical plant to the one that's producing the crop that you really are interested in. And so that this is one reason that cuttings are really important sort of from an agricultural perspective. So we can take this cutting business even further. And one of the things that is surprising, because now it doesn't look like it should propagate, is that leaf cuttings can be made such that the entire plant reproduces from a single leaf. And so going back to our snake plant, we have our two methods by which snake plants are propagated. You can see that a single leaf can be taken off and a V sort of cut made to increase surface area and put in water, and that will root. But snake plants can also be just, this leaf can be directly placed into soil, and that will also produce new snake plants. So you can see that plants are quite resilient in their ability to propagate and survive quite a lot of trauma. And the reason these plants, and a lot of times you'll see this take place with cuttings, the leaf has been cut at the top, is to reduce that surface area for water loss. So when we put this leaf right into the soil, and soil more so than water, this top part of the leaf will heal, but what we're really trying to do is reduce the amount of water that's being lost through the leaves as the plant regrows its roots and is able to then access more water. So let me just give you a sort of a fun example of leaf cutting with begonia plants. And so with begonia plants, you can take a leaf from the parental plant, and what you do is you flip the leaf over, and then you with a razor, like an X-Acto knife or a sharp paring knife, you cut some of these main veins. So if you look at the top right picture here, you can see that they're veins that run from where the petiole is, and you cut the underside, and then you just put the leaf on moist soil, and you use a few pins, like they look like essentially little staplers, and you hold the leaf down to the surface. And over time, what you'll find is that original leaf will wilt, but in its place where the wounds were made, you'll find both shoots and roots of multiple begonia plants starting to form. And you might need to modify this a little bit based on how dry your environment is. So you can put saran wrap on loosely just to reduce that moisture loss, because a lot of times with plants, what we want is some level of moisture in the soil, but we're also always worried about having too much moisture that promotes fungal and bacterial growth. Succulents, I think, are often people's favorite plants, and they're also incredibly easy overall to propagate. And so here you have like a Christmas cactus on the top left, a jade plant on the bottom left, as well as sort of a puncha cactus on the bottom right. And with the Christmas cactus, these entire stems are actually one leaf. You can just break them off, and you can just directly plant them into soil and or water as before, and they will essentially establish quite readily. With jade plants, there are multiple ways you can approach this. Of course, with the Christmas cactus, you can break it up and split the plant too. But you can start with stems of jade plants or individual leaves. And if you place individual leaves on the soil, what you'll find is that they'll quickly root and also start forming new shoots at the location that are contacting the soil. So this is something that succulents are particularly good at because they don't dry out very readily. One thing that you do differently with succulents from let's say the begonia, which is quite thin and not waxy, is that sometimes people like to just leave the succulent. So if you break off the jade leaves, you just leave them out for a couple days, let that wound heal completely by drying out, and then you put it on the soil. And that's something you can do with these apuntia, where you break off a pad, you just leave it out for maybe a couple days so that the broken off exposed tissue dries out, heals. Of course, you're going to be using leather gloves or tongs or something to handle apuntia, and then you just place that right into the soil, and it will usually like a cactus mix, and it will reform into a new plant. The last thing I'd like to talk about, just because it's I think we get into air layering, it's fascinating, is the idea of layering. And so here, we're propagating plants without doing the cut first. And so going back to our spider plant, so rather than pulling off the plantlets or the spider plant babies and putting them into soil or water, what we're doing is we're just putting that plantlet while still attached to the parent plant right into soil. So the downside of this is that you end up taking up a lot of space if you want to do a bunch of plantlets at once, sort of like an island of spider plants. But the upside is that because the plantlet is never pulled off of its parent, there's no trauma. And people have found that the plantlets grow sort of quicker while still attached to the parent, because it's still able to access some water and nutrients from the parent. And so once the plantlet has established, then you would take a pair of scissors or whatever, and you just cut the attachment off, just like you'd sort of cut like the umbilical cord essentially. And what you can also do is sort of have serpentine layering, where you have sort of multiple pots along the same stem, where the plantlets, like the multiple plantlets, are all in their own pots, and so it looks like a snake because you have all these plantlets in a row. But this is one nice way of propagating plants, particularly things like with runners, like strawberry plants. If you want to grow extra ones and move them elsewhere, this is a nice way you could do this outside and then just move the established plant elsewhere. But one of the more fascinating kinds of propagation is air layering. And in air layering, we're kind of doing something very similar to what we just saw with the layering, but it's not at ground level. Rather, it is directly on a plant, like a chestnut or an apple tree or whatever, or roses. And the idea here is to get roots to form right above ground, right on the plant. So you can see in this 1903 drawing, there's a pot right on the stem, and there are roots growing right into that pot, such that you can cut under the pot and then move that plant elsewhere. And you'd still have the original plant, so the original stem at the bottom, plus a second plant that is now rooting in the pot. And there are little fancy devices where you can do this, but air layering actually is not that complicated. So let's look at one example. So I have a rubber tree. It's not this rubber tree, but one that has a problem that it grows really tall, and it's sort of at some point is probably well, it's almost like 2 meters now. So it's kind of just like very unstable and um, gawky. So how would you maybe cut that plant down to size, but also propagate the rubber plant? And so the key here is that we expose part of the woody material of the plant stem. So what we're seeing here on the left-hand side is the rubber plant. And what we're seeing in the middle picture is somebody cutting the cambium, sort of like the bark and cambium off the stem and exposing about somewhere between 3/4 and 1 inch of this wood, sort of very close to a node. A node is the point at which the leaves form. And so you pull all the leaves below this cut and maybe 1 or 2 leaves above the cut. You let the cut heal like for a day or so, so it dries out. Then you apply rooting hormone to this upper part of the cut. And the idea here is to get roots that come, I'm just going to draw here for a sec. You want roots that come growing from this part of the surface, which is pretty incredible. Now, the essential part is that you cut through the cambium, because cambium is sort of is growth tissue, and it can heal over the wound and sort of reconnect the upper and lower parts of this this cut. But if you let that be exposed, then what you do is actually relatively simple. You don't have to use sphagnum moss or something, but a lot of people use moss because moss has the property of being able to retain moisture at the same time not get infected. It has sort of natural antibiotic properties. So you soak some moss, you wrap it around that cut after you've put the rooting hormone on, wrap a paper or a plastic bag or saran wrap or aluminum foil around the moss just to hold it there and keep it relatively enclosed. You have to periodically open it you have to open it periodically to add moisture, but over time, what you see is that there's root formation. So on the right-hand side here, we see root right around the main stem there. And then if we were to take the plastic bag off and if we were to snip just underneath where this root ball is, you can see that that root formed right on that stem, which is obviously easy to transplant into a pot. And so this is a way, you can do this outside, where you can propagate your rose plants, just one way, or your apple plant if you have a particularly favored apple tree. This is, it's kind of fun because it just seems so strange to see these little packages on a plant, and then it becomes a means of creating another plant. So at this point, I hope you can appreciate that it's not necessarily that complicated to propagate your own plants. It's really sort of a hobby for me, but it also is a way to sort of just have a chance to look at some of the amazing biology that takes place with plants. It also, when you start propagating, there's a great community around that you can trade plants and so forth. And so that's something that I hope you have a chance to experiment with. So thank you so much for spending this time with us. And just to let you know, we're having one more webinar on making terrariums in December on the 5th. And if you just want to get notices and so forth about these workshops, just please sign up. Thank you so much, and I'll stick around and answer as many questions as I can.
Awesome. Thank you so much, Tan. I see we do have a couple of questions coming in about recordings. We have recorded this session and we'll be emailing that out early next week, along with, I believe, some slides as well and some information about the next webinar as well, with a link and everything, so you can sign up there. Yeah, we got a lot of questions coming in, which is fantastic. Again, if we are not able to get to them right away here, you can email us at scemarketing@MacEwan.ca, just on the screen there, and we will try to get all those questions answered through that channel as well. So to get right into the questions, because I know we're running late on time, one of the first ones we had in came from Patty, just wondering about the sun-grow mix that you had in one of the beginning slides there. Sort of what is it made up of? What is the composition? And are there any good soils that you would recommend for propagating plants?
I think that there are many options, and the only reason I chose that one is I think that it's a company that's based out of Wabaman, and so it's a brand that I've used for research work as well as just for houseplants. You can find it at Apache Seed or Canadian Tire. It's pretty readily available in Alberta. So the thing about choosing the soil is, I think it's important to think about the kind of the paternal plant, the original parent plant is, and what it likes to grow in. So if you think about something like a cactus mix that's relatively coarse, stays relatively dry, that doesn't hold and retain water, is something that you would transplant your little nubs of cactus into. Whereas something that is maybe something that is more tropical, where the plant needs sort of a more consistent level of soil moisture, then you'd want something that maybe with more moss or more a way of like retaining a little bit more moisture in the soil. Having said that, in almost every case, you want soil that's not too compacted, because that will lead to problems with growth as well as the inability to dry properly. And so the Sunshine #1 mix is like it's a pretty basic mix with moss and vermiculite and topsoil like stuff. And so it's generically good, but not necessarily ideal for every kind of plant. People often like to use sort of like rock wool or cocoa coir, like just sort of other media. Sometimes if they want to have more control, like maybe in an industrial setting. And yeah, so there's lots of choice. I think that my thought a lot of times is if I just get it going in water, like so not necessarily with succulents, but with the other kind of cuttings we're talking about, then we can just I just transplant into a generic soil mix after. So that could be one advantage of using water as a growth medium first, like a hydroponic setup, before you transfer into soil. But I think you could tell by the length of my answer, there's lots of options and choices.
Awesome, thanks for that. And then we did have quite a few questions come in when you were talking about transplanting. So one of the things that came up a couple of times is how can you tell when they need to be transplanted, sort of what size pot, how you can tell what size pot, or if there's like a minimum amount of time you should go between transplanting. So
I think one of the things that's always surprising to me is how little transplanting you necessarily have to do. It can seem very snug for the plant, but for example, the rubber plant I had for the longest time had maybe like a 3-inch pot, even though it was 1.5 meters tall. So it's kind of the real problem at that point was that it was tipping over because there was not enough mass at the bottom. I think that as you're sort of thinking about the space you have in your home, so this is more like an aesthetic choice. Yeah, I always like to imagine like how big I want the plant and where I want the plant to be. Like, of course, in an ideal setting where the plant's doing well and everything, because I think a plant would always love to have like a bigger pot more soil, more light, more resources, but I may not want a plant that is that tall, because a lot of plants, one of the things about a lot of plants is that they have indeterminate growth. They grow as long as they're alive, and a lot of house plants tend to be sort of tropical understory plants, because it's sort of a low light environment that we have indoors typically. So in the tropics, sort of underneath the really big trees, it's actually quite dark, right? Because of all the canopy that's blocking the sunlight. And so those conditions are not that dissimilar from the plants that we have here. So, indoors. And so the problem is then, like, these plants can get enormously large without sort of adding some sort of constraint. So it's a great thing when plants get big, but my place is not big enough for plants getting to their big size. Yeah.
Awesome. Yeah. And sort of like along those lines, with the transplanting, knowing when to transplant, is there a way that you can tell, like, when the plants are ready to be propagated? And also, are there any plants that you can think of that maybe don't do very well with propagating or are really hard to propagate?
The reason I brought up orchids, I'm going to answer that last bit first, is that I think that orchids sort of in the plant world are sort of revered for how hard they are to propagate sometimes. And so I guess I would say that is sort of a pretty specialized plant. And there's an orchid society in Edmonton that they're like just an incredible concentration of very serious orchid plant lovers. And so, and part of that is the challenge of trying to propagate as well as just even grow some of these orchids. And so one of the things about orchids is that, this is sort of an aside, but they don't tend to establish, their seeds are not laden with energy. They need to form fungal associations to germinate and form plants. And so it's they're just hard, I guess, for different reasons, and they're relatively slow growing. I can't remember the first part of the question already.
They're just wondering if there's like a good rule of thumb for when a plant would be ready to propagate, like, or if you need to avoid doing it too early or anything like that.
Oh, I see. Yeah. I mean, sort of the bigger, the healthier the plant. That's a rule of thumb. Like, the better your chance of success. And if it's too small, and you can certainly split it, but it sort of just logically, there's not enough plant material to serve as sort of a buffer. So I think, like, particularly right after propagation, like, let's say you take a cutting and then you propagate it, you probably want to give the plant like quite a bit of time to establish its own root system and grow a bit. Having said that, like, I think that it's really sort of for me, like, just a challenge and sort of an interest to try to test different ideas and things about these plants. And so like, if I were feeling experimental, then I might just start with a big plant, propagate lots of small ones, and then I can run sort of propagation experiments on those little plants without fear of losing my original plant. So I think it starts to become like a game of multiple years and sort of like a lifetime sort of spent on growing plants and propagating them. But certainly, I don't think there's necessarily a limit to when you can propagate it. Just typically, the less resourced something is, the higher the risk and the more care you'd have to spend to make sure you're successful, I guess, in propagation.
Yeah, I know I recently started propagating my own plants for the first time. It's definitely addicting. Once you get like the first success, then you just can't stop.
It is.
Yeah.
Yeah.
We are coming up to 7 o'clock. I don't know if we have time for a couple more questions before we sign off for the night.
Sure, I'm happy to. Yeah.
Awesome. So we did have a couple questions come in about how well these methods might transfer over towards propagating like outdoor trees or outdoor shrubbery. Sort of, will it work? And like, if you're going to go about it, is there a general rule for like how deep or wide you should dig around the roots for your best chances of success?
So my first reaction to that is like when I think about outdoor, for the most part, not like for small animals, like small plants, I guess, but overall, I feel like just everything gets supersized. And so when you're talking about splitting sort of like a hedge or like a bushy plant, then you're not talking about just being able to work with your hands. Like it becomes actually quite manual laborish to dig around a plant, to have the strength to actually break up a root ball and so forth. So the principles are exactly the same, just that our outdoor plants tend to be larger than our indoor plants. I guess that's not necessarily true for potted plants and sort of container plants in general. So container plants, I would say, are identical, but I think something that I often am always surprised by is like how deep certain root plants are. I think with a lot of outdoor plants, I find that there is more of an opportunity to try to harvest seed. But of course, then you're starting sort of with like a very tiny plant the next year. So, but I think that that's a great way to really, if you really want to have lots of plants, it's nice to wait till the end of summer or sort of early fall, collect those seeds as they mature, and then save them for the next spring. A lot of times with plants that do well in Edmonton, you need to treat them in a fridge or with moisture. So like you're sort of trying to mimic that that the winter conditions that trigger sort of metabolic processes in a plant. And so if I were to want to propagate sort of especially native plants, I would collect the seeds in the fall, but then about 2 months before I wanted to start thinking about maybe germinating them in trays, I would put them in a fridge with sort of moist paper towel, leave them in there for maybe 2 weeks, and then I would sort of try to germinate them, because that helps the success rate of seeds germinating. So other than that, like, yeah, like I think the thing is you can, with the air layering, for example, that's something you can do with trees, like apple trees and pretty large rose bushes. Like they actually do better because they can support the weight of the moss, and you don't actually even have to use moss. Like some people use sterile soil, and so yeah, I think that that's all these ideas apply. Just, you know, you need shovels and hose and like picks and things like that to try to work on some of these plants and so forth. Yeah.
Awesome. We also had a question come in the Q&A, wondering if you could maybe just explain a little bit more about what the hormone rooting powder is, or if there's like a natural equivalent. I know that I saw somewhere like you can possibly use cinnamon. That might just be a wives' tale though. So if you have anything, sort of any knowledge in that area for us.
Yeah, I mean, I have not personally tested a lot of these methods. Like I've heard like aloe vera, like there are obviously natural auxins. All plants have auxins. Sorry, so I should step back. So in the case of the rooting powder, it is an auxin, which all plants produce, and what auxins do typically is enhance the growth rate of cells. And the kind that you buy is sort of a synthetic version of a natural auxin. So just like Lauren was saying, there are certainly going to be options that are natural. I just, like, for me, I guess the main thing is that because you're exposing sort of like a cut surface of the plant to the substance that enhances the growth, I worry mostly about infection and sort of introducing something that will damage the plant. And so the powder I find is like just very shelf stable. And what it does is when you dip the powder on, it mimics, it's sort of like, so the plant would naturally produce those auxins in cells. So a plant is fascinating because it's very modular. It doesn't have a nervous system like we do, so it can't necessarily communicate sort of like your thoughts to your hands moving or whatever. But what it does is it transfers chemicals from cell to cell to cell, and part of that communication system are these hormones, and auxins are one of them. And so a lot of this stuff will absolutely work with no hormone. And so I think that's just probably a crutch I use because I have it available. I think, like, for example, with the hibiscus plant that I talked about, you certainly can get that hibiscus to germinate, or sorry, not germinate, set root in water, but it just might take longer. And the problem with it taking longer is then you have to maintain it, that water sterile for a longer period of time. And so it just sort of amplifies the degree of difficulty, but it certainly, if you don't want to use the product, in most cases, you would not have to use it at all. So yeah, it's just an external source of hormone to mimic the natural process.
Awesome. Great. I think maybe one more question might have time for tonight. Sure. I know some people were asking about different propagation methods for very specific plants. Probably don't have time to go through a list tonight, but do you have any tips for like telling what type of method to use, or knowing which plant like you could propagate from a leaf versus one you might need to grow from the node for?
So unless it's like a very high stakes situation, what I really want to encourage everybody to do is just to try it, because that's one of the things that always surprises me and excites me, is when I just take a leaf, put it in something, and it's rooting, and you're off to the races. Certainly, you can, in one of the great things is this community of plant growers is very tightly knit and very enthusiastic. So you could pretty much find specific information about any plant that you can identify. So I guess if you have an unknown plant, then that might be a challenge, but you can also post pictures of your unknown plant, and somebody can help you identify the plant. But overall, like, I guess what how I like to sort of explore my own curiosity is sort of just like by looking at a plant, thinking about how it might be growing in its natural environment, right? And sort of trying to take a guess as to how it would propagate clonally. So, like, of course, like, there's always the sort of brute force method of trying to get something to flower and pollinate it and set seed. But on the other hand, like, virtually every plant grows and propagates asexually as well. And so I think that's sort of trying to like guess based on the structures that you see on the plant, like sort of, I guess if you're to expose the root, sort of just see where natural breaks are. But I think you'd also be surprised that if you focus on the areas of the plant where there is this meristematic tissue, for example, near the nodes or at the apex, the top of the plant or so forth, and also the root, you can have a pretty good sense of where the plant would try to regrow if you were to give it the opportunity to do so.
Awesome. Well, thank you so much for spending the time with us tonight for the presentation and that little bit of extra time for the questions. I think that does probably wrap up our time for today. We had a very big group, so we didn't get to probably about half the questions we got. So again, please feel free to send us an email at that scemarketing@MacEwan.ca, and we are more than happy to get more to your questions that way. And again, we did record the session as well, so you can look forward to that email sometime next week. Tan did already mention the webinar that we'll be having in December, so it's going to be about building your own terrarium. We'll include a registration link for that in the email that we send out next week. Or of course, you can always find it on our Facebook events page, which is at MacEwan.ca, or you can follow us on any of our social medias for updates about anything going on at the School of Continuing Education. So big thank you again to Tan and everyone that was able to attend tonight. I hope everyone enjoys the rest of their night, their weekend, and some of this nice fall weather that we're having before the snow flies, which undoubtedly will hit us sooner than we think. So gonna appreciate the weather for now.
Thank you so much, everybody. Really appreciate that you decided to spend time with us this evening. And thank you, Lauren, for making everything go so well.
Anytime, and thank you again.